What we're going to do in this video is provide an overview of what porifera are and how they deal with gas processing. Here we have a photo of a porifera. Here we have a photo of a porifera, also known as a sponge. And here we have a diagram of one near it, next to it. There's also a diagram of a cell that the porifera commonly has, called the choanocyte, lowered, which we'll get to later. Now, you may be familiar with the notion of needing oxygen to survive, and these sponges are no different. Both our cells and the oxygen cells need oxygen molecules to survive. And what was unique about these sponges is that they don't have complex respiratory mechanisms like us human beings do. Instead, the sponges use cells called choanocytes, which are which are located on the inner chambers of the sponge. So that would be somewhere here. Inner chambers. And the choanocytes have many unique features that help the sponge, but the main one we're concerned with for this process is this tail bit here, called the flagellum. And the flagellum is surrounded by these small hair-like threads called the microvilli. And the flagellum's job is to be moved back and forth by the cell and in a rhythmic motion, which draws a water current towards the cell. Let me just switch to a different color there. Which draws a water current towards this cell and thus towards the sponge. The microvilli help support the cell and maintain the stability of the choanocyte while the flagellum is beating. The water current, drawn in by the choanocytes, will enter through this opening here called the ostia. And it's a small pore in the covering, and the water can in a controlled rate can enter the ostia through the ostia in a controlled rate. It passes through a system of canals in the sponge, and as it does that, it passes through other cells like other choanocytes or other types of cells like amebiocytes, which do other things like transport nutrients, and we'll discuss them more later. And as they move past the cells, the oxygen molecules will diffuse from the water into the sponge. From the water into the sponge cells. Because the water has a higher concentration of oxygen than the cells, through simple diffusion, the oxygen will go from the higher concentration to the lower concentration, and since it's small and non-polar, it can easily move through the cell membrane. Likewise, any CO2 from inside the cells will go back and will diffuse into the water because there's a higher concentration of CO2 inside the cells than there is in the water. And the water will keep flowing past the cells as this happens, and it slowly collects all the CO2, and then finally it enters this opening here called the spongosoal. It's a large central cavity, and the sponge has spongosoal, and from there the water will get expelled. The water will get expelled through this gap here called the osculum. And having this gap here makes sure the sponge doesn't have it doesn't collapse under the pressure of the water from inside the cell trying to get out. And as the water gets expelled, so does the carbon dioxide and any other molecules that it picked up along the way. We'll talk more about this process in further videos.